We've been promising all the earlier new data, and I get to deliver it now. So I'm going to talk about our newer clinical trials, and I'm going to talk about other applications for the RNS system relevant to questions that were asked before the break. So we are in clinical discovery mode in two areas. One is to collect data to expand indications for use. I'll share with you the primary efficacy endpoints for the post-approval study. You know about the Nautilus study. NIH has funded a study in a very devastating symptomatic generalized epilepsy, Lennox-Gastaut. I'll share that with you. And exciting news about a true collaboration and partnership with FDA to understand how to use the real world data that is accumulated in pediatrics. We'll also talk about what AI is doing. We invested in AI before it was popular, over 5 or 6 years ago, understanding that our database was so massive that there was no way we could figure it out. We are finding objective neurobiomarkers, and we're using those biomarkers to be really smart about programming, detection, and stimulation, and also tracking the patient's response. So the post-approval study is the largest FDA-required prospective post-approval study. It is a really tight study design. This is for patients newly implanted with the RNS system. The first enrollment being in 2015 and the final implant 5 years later. We have the final patient to 3 years, so we've been able to look at the primary effectiveness endpoint submitted to FDA, and it was accepted in December. We will be presenting the results at the American Academy of Neurology in the spring, but you're going to hear them first. So let me talk about the patients who enrolled in this trial. 271 of them stayed in the trial for 3 years, which is pretty impressive because they were living their lives and moving and starting new jobs, and they stayed in the study. For context, the average frequency of seizures when patients enrolled was 5.7 per month. Similar to the pivotal trial, we had a majority of patients who had more than one focus. We had patients with seizures everywhere, neocortex and also mesial temporal lobe, and then a number of patients who had onsets in both areas. We found that about a third of patients did not have intracranial monitoring before they were treated in the RNS system, similar to our pivotal study. But here's a big difference. And I talked about RNS moving earlier in the treatment paradigm. So whereas in the pivotal trial, a third had been treated with a prior epilepsy surgery, largely resection, that's down to less than 20% in the post-approval study. And look at the drop in use of vagus nerve stimulator. 32% of patients had been treated with a VNS, and in our more recent study, it's down to under 8%. Here are the results. We have data out to 3 years, and I'm showing it to you in 6-month increments. You'll see the sample size, and in purple, you'll see the median percent change. In parentheses is the response of the upper quartile and the response of the lower quartile. You will recognize that we're seeing very impressive responses really early in the course of treatment. And we're seeing that those improvements accelerate pretty quickly. In the upper quartile, we have over 90% reduction, with 42% of patients seizure-free for at least 6 months, and we're up to nearly a quarter of patients who are seizure-free for at least 1 year. For the lower quartile, I am so optimistic about the opportunity to drive this further, given our continuing clinical experience and the value of the AI. But getting to this flexible placement and benefit everywhere, the dark blue is the improvement in all patients, and you'll see in the filled in blue, the patients with actually the open blue, patients with mesial temporal epilepsy, the filled in blue, neocortical patients, and stippled patients with onsets in both areas. And so it is clear that the RNS system works for anyone with focal epilepsy. So let's contrast this, and I'm going to talk about Medtronic's registry for epilepsy, and that's using DBS. These are not comparator, head-to-head comparator studies, but it is a similar patient population, prospective, and they also had retrospective data. All of our data was prospective. But the difference is, and this is peer-reviewed literature for the MORE registry, are pretty striking. That the median percent reduction from year 1 to year 3 is not what we see with the RNS system. And this represents current contemporary medical practice. These publications on the MORE registry are very recent. They're from 2024. So did I skip ahead? No, there we go. So what can I say in summary about this? Well, we continue to see that efficacy improves over time, and we have a home run in a substantial number of patients. 90% reductions within 6 months are possible, and seizure freedom at 3 years. Why are things better? Well, you have a sense of all that we have learned. We have learned what should be detected. When we started out, we waited until patients were well into their seizure in order to intervene. And now we recognize that we aren't, it doesn't work best if you do a seizure, if you do use it as a seizure stopper. It works better as a neuromodulation device. We understand better where to put the leads and where to provide stimulation. And we know where to start with the stimulation programming from the very beginning. And we know how to fine tune based on what the patient comes in to tell us. When we say, how are you doing? They describe that. So we use that. That's really important. But we also have the biomarkers, and we understand how to move forward with treating according to biomarkers. Now, I told you we haven't forgotten about the 15% of children who have focal epilepsy. And we, some years ago, started a response study that really shows our commitment to advancing our indication for use to a pediatric population. FDA required a prospective open label trial. And frankly, in contrast to any study NeuroPace has ever done, we've had challenges in enrollment. And this is very typical for any company that's trying to do a pediatric study for a therapy that is widely available to adults and for which physicians are providing it according to the practice of medicine, not everywhere, but some. So in the meantime, people are publishing all these publications about patients treated, children treated with epilepsy. And we became aware of a database, Pediatric Epilepsy Research Consortium, a 10-year-old database or 10-year-long database of children treated according to the practice of medicine across 27 pediatric epilepsy centers. So when we put that together and we add to it support from advocacy groups, in fact, a demand that children have official access to this, we were able to partner with NEST, the National Evaluation System for Healthcare Technology, that is funded by FDA in order to promote the use of real-world data to expand indication for use, particularly to underserved population. And we are a perfect project for NEST. And as part of this, we are able to collaborate with FDA with several meetings per month, sometimes with over 25 individuals from FDA in attendance, to discuss how we can use all of these data sources to support an expansion for indication for use. We are well along in this process. NEST uses a group of epidemiologists. There's a set process by which this is done, which includes looking at the data sets and understanding whether they contain data points that would that FDA would be able to accept, and design then a fit-for-purpose study to access that data set, including a feasibility and a concept protocol, which is where we are right now. We are getting very close to the official NEST mark of approval. And what that means is FDA has agreed in principle that the data is available and that they will accept that as sufficient. We then execute on that. We implement, we do data analyses along with NEST and provide that data to FDA. It has moved along since the data already exists. We believe that this will go very quickly and that we will be able to submit in the second half of this year. Now, there's some interest in Nautilus, idiopathic generalized epilepsy. I mentioned this epilepsy before and told you that this happens in otherwise neurologically normal individuals. Medication is the only option. There is not an option for surgery. The risk for sudden unexpected death in epilepsy is particularly high in this population because these patients have generalized tonic-clonic seizures, which are the seizures that put people at particular risk. We, based on the literature, elected to provide treatment in the central median nucleus of the thalamus. We are moving ahead with an indication for use as an adjunctive therapy. FDA allowed us to enroll 12 years of age and up. This is a randomized controlled trial, prospective, multi-center, sham stimulation controlled. And Mark Richardson alluded to how rapid the enrollment was. 100 participants enrolled quickly, 87 implanted across 23 centers. The image to your right is pretty profound. We didn't know whether we could detect seizures in the thalamus. And so what you see right there is the RNS system detecting a seizure in the thalamus, really establishing the feasibility of this approach. Here is the timeline. By providing the publications and experience to FDA, we were granted a breakthrough designation. Many or not all of, or probably all of you understand that this really speeds up the regulatory path. Our first enrollment was in 2022. Our last enrollment was within 18 months. That's a year faster than I had predicted, really speaking to the interest and need. The final patient will complete the 1 year endpoint, which is when we look at our primary effectiveness and safety. And I would love to be able to provide those results in March of 2025, but there's a whole lot of work that happens after the last visit to the time that we are able to submit to FDA. And I've listed some of that work. I will just, in the interest of time, tell you it is a great deal. However, we're optimistic that we will be ready to submit to FDA in the second half of 2025, about the time that we submit for pediatrics. We will continue to follow these patients to 2 years, but the primary endpoints are at that 1 year time. We mentioned Lennox-Gastaut. This is the worst type of epilepsy. It happens in childhood, and parents are faced with a child who develops progressive encephalopathy, really deteriorates in front of their eyes and has multiple daily seizures. We were able to receive a very sizable grant from National Institutes of Health to fund this entire study. It is being conducted at 6 clinical sites, and in addition at a site that is in charge of the neuroimaging part of this study. It is also, this study is funding biomarker investigation. We have all 20 patients are implanted. Safety is very acceptable at this point. This was a 5-year grant. The grant completes 2026. And at the end of that, we will have a very good idea about safety, which is the primary endpoint in this feasibility study. We'll have information about efficacy. We'll understand about biomarkers that we can use to drive treatment in this patient population. And we will know ahead of time about where to put the leads using computational methods that we are developing as part of this effort. So I want to tell you about all the data that we have. We have over 6,500 patients that are being treated, and it's like an oil well. Every day there is more data. This slide is out of date every time I show it. There's over 19 million stored samples of intracranial EEG. And what we have samples when the patient is at baseline, meaning that when we look at it, we don't see anything happening. Samples when there are epileptiform discharges, and we have samples when patients have electrographic seizures. I will tell you that what we discovered is each one of these types of intracranial EEGs are telling us something. And the algorithms we have developed are giving us biomarkers that correlate with patient-reported seizures. And those biomarkers are what are going to drive our innovation in the application of the existing technology. And I'll just show you one example. Treat to clinical seizures, let's treat to biomarkers. And so what we started out is using machine learning tools to look at. In the top panel, you'll see year 1 to year 7 in individuals with mesial temporal lobe epilepsy. The x-axis is the frequency of epileptiform spikes. The y-axis is a frequency spectrum, or the frequency domain. In the top row, you see patients who did really well. And in the bottom row is patients who didn't do well. They stayed, a 25% reduction. What we see is that everybody started out the same way in frequency and epileptiform spikes, but as patients got better and better and better, they had fewer epileptiform spikes. This tells us that we should treat epileptiform spikes. We then developed deep learning capabilities, and now we're using vision transformers very recently to understand whether we could go deep and identify those patients who are going to be responders and those who are having a less robust response. And it was very clear that going down 50 levels in this analysis, which was using a convolutional neural network, that it very clearly distinguished that there are differences in these patients. So this is another window to the brain. Well, can we use this data in general? It has some value. It has value to us because of all of the information it provides us about patients. These are my 2 patients. And where you see the little round M, that's the patient swiping a magnet to tell me that she has had a seizure. And the green bars are when I actually see seizures. So you see that when this patient tells me she's having a seizure, she is, but she's not able to tell me about all of them. But it's a pretty good indicator. The patient on the bottom is a patient that has been so hard to treat because she has no idea when she's having seizures. And I've been treating her for 20 years blindly. With the RNS system, I now know when she has seizures that she doesn't know about. And in January 2022, I found better detection stimulation programming. And now when she has them, it's when she decides she's not going to take her medication. So what is our data telling us? Well, we look at different types of data. And in the upper left panel, you'll see one intracranial EEG that's showing that the patient is having prolonged epileptiform activity. It's not a seizure, but the device is detecting a number of abnormalities. To the right, you see that there is an electrographic seizure. There's a buildup in these discharges. The patient doesn't know that this is a seizure. And then the bottom panel is an electrographic seizure that the patient has symptoms with, so the patient knows. We have looked at all of these types of recordings and have found that those long episodes, upper left panel, are strongly correlated with the patient's clinical experience. And this is an example then how this can be applied to drugs. So to the left, you'll see the spike rate, that's the middle line, for a patient who's being treated with the RNS system and was started with a new anti-seizure medication. And we could tell, as is demonstrated in the graph to your right, within 1 to 2 weeks, whether that drug was going to work or not. That relieves patients of the burden of drug trials that can go on for 6 to 12 months. But it also was very interesting to a company called Rapport. This is a Janssen spinoff. It's a company with a very interesting compound that binds specifically to proteins on the AMPA receptor that are very prevalent in the mesial temporal lobe. Well, guess who has a lot of patients with mesial temporal lobe epilepsy? Now, this company wanted to perform the very first study in people to see if there is target engagement, and based on that experience to make the decision about whether to invest in clinical development. They came to us and said, we understand from your publications that long episodes are biomarkers for patient clinical response, and we want your long episodes to be the endpoint of our study. This is a whole new paradigm in very early drug development.